Welcome viewers. Today I will be talking on vitamin A, vitamin E and vitamin K. These three vitamins are otherwise unrelated except that they are fat soluble, and we will see that they have some sort of structural similarities. Let's start with vitamin A. The discovery of vitamin A roots back to 1912 when Frederick Hopkins demonstrated that there are some quote-unquote accessory factors present in milk other than protein, lipid and carbohydrate, which is necessary for growth in rats. These accessory factors were termed as fat soluble in 1918, and in 1920 they were named as vitamin A. In 1919, Harry Steenbock proposed a relationship between yellow colored plant pigment, which is beta carotene, and vitamin A. In 1931, Swiss chemist Paul Carrer described the chemical structure of vitamin A. And vitamin A was first chemically synthesized by two Dutch chemists, David Adrian van Dorp and Joseph Ferdinand Arens. Meanwhile, in 1929, Frederick Goland Hopkins received Nobel Prize in Physiology or Medicine for his work on vitamins. In human diet, vitamin A comes in two forms. The preformed vitamin A, which comes mostly from food of animal origin, contains retinol and its esterified form. And the provitamin A carotenoids, which mostly come through plant products, gets converted to vitamin A within our body, and these are mostly comprised of alpha carotene and beta cryptoxanthin. If you look at the structure of vitamin A, which are retinol, retinal, retinoic acid and beta carotene, we see that all these contain a beta ion ring and an isoprenoid side chain. There are some carotenoids which are characterized as the non-vitamin A carotenoids, which does not show any vitamin A activity, like the lycopene, which is present as bright red carotenoid pigments in tomato, red bell pepper, watermelons and papayas, or the luteins, which is a xanthophyll, but they do not have any biological activity, and we can see that the characteristic beta ion ring is not present in their structure. This slide shows the conversion of the provitamin A carotenoid pigments to the biologically active form of vitamin A, like retinal or further retinol, by the action of 15' dioxygenase enzyme present in our body. Food rich in vitamin A include animal products like fish, egg, meat and dairy products, and plant products like green leafy vegetables and fruits and vegetables that are intensely colored, like bell peppers, carrots and so on. This table shows the reference intake level and the upper limit of vitamin A. It is important to note that vitamin A is a fat soluble vitamin, and hence its urinary excretion is poor. So if vitamin A is taken over and above the required level, then there is a possibility that vitamin A will accumulate in the body, and if that crosses a particular level, then toxicity symptoms may arise. Thus, there is reference daily level as well as an upper level for vitamin A. For infants, it is 500 microgram per day and the upper limit is 600 microgram per day. For children, it is 400 and 900 microgram. For males between age 9 to 70 years, the RDA varies between 600 to 900 microgram per day and the upper limit is 1700 to 3000 microgram per day. The highest being for pregnant and lactating women, which is up to 1300 microgram per day, and the upper intake level is set at 3000 microgram per day. The global scenario of vitamin A deficiency is very alarming. This picture shows that inhabitants of the entire of the Indian subcontinent and the continent of Africa are severely affected by vitamin A deficiency, and the status of deficiency is clinical. The major deficiency symptom associated with vitamin A deficiency is xerophthalmia, which is a condition where the eye fails to produce tears and become dry. Children and pregnant women are specially susceptible to vitamin A deficiency, and the early symptoms of xerophthalmia is night blindness, where an individual feels problem to see in low light. A photograph is shown which is for the normal vision, and the photograph next to it is the type one sees when he or she suffers from night blindness. The major function of vitamin A is its role in vision. The rod cells present in the retina is packed with photosensitive proteins called rhodopsin. The two major components of rhodopsin are the retinal and the opsin. Retinal is vitamin A and the opsin is the protein. In rhodopsin, the retinal is in 11 cis retinal conformation, and when light falls on rhodopsin, there is a conformation change in this retinal molecule, and the 11 cis retinal gets changed to all trans retinal. And all trans retinal does not stay bound to the opsin, it dissociates out, and this dissociation of retinal from opsin is actually the nervous signal which propagates along the optic nerve and reaches the brain, and the individual gets the perception of vision. Another important function of vitamin A is in the form of retinoic acid, and it acts as a generalized morphogen. Retinoic acid diffuses through the plasma membrane and reaches the cytoplasm, where it binds to its receptor called the retinoic acid receptor. Retinoic acid receptor bound to retinoic acid gets entry through the nuclear pore complex, and binding to these rare elements, retinoic acid controls transcription of many genes that are responsible for normal growth and development in different forms and stages of life. In 1938, Weidenbach first demonstrated that when wheat germ oil is supplemented to 17 newborn infants who were suffering from growth failure, 11 of them resumed their normal growth. In 1946, a research team led by two brothers of Ontario, Canada, namely Ivan Schutt and Wilfred Schutt, they showed that alpha tocopherol improved impaired capillary permeability and low platelet count in experimental and clinical thrombocytopenic purpura. In 1949, Jarlozzi administered all-rack tocopherol acetate to prevent and cure edema. And since then, from 1960s, supplement of infant formulas of vitamin E to newborns became a routine practice in almost all parts of the globe to prevent hemolytic anemia. Structurally, vitamin E has 8 vitamins. These are named as the alpha, beta, gamma and delta tocopherol and alpha, beta, gamma and delta tocotrienols, of which alpha tocopherol is physiologically most important. Food rich in vitamin E are cold preserved plant oils, apricots, colored bell peppers, almonds, green olive, papaya, avocado, cooked spinach, sunflower seeds and so on. And the dietary reference intake level is 4 mg per day for newborn to 15 mg per day for 14 years or above age. Deficiency symptoms of vitamin E includes peripheral neuropathy, ataxia, skeletal myopathy, retinopathy, and impairment in immune response. People who are suffering from Crohn's disease, cystic fibrosis, or have an inability to secrete bile are at high risk of vitamin E deficiency. This is simply because of the fact that in these conditions, absorption of fat in the intestine is severely impaired, and since vitamin E is a fat soluble vitamin, along with impairment of fat absorption, vitamin E absorption also goes down and they develop the symptoms of vitamin E deficiency. A major function of vitamin E is to act as an antioxidant. Reactive oxygen species, or the ROS, are produced in the body of all aerobic organisms as a consequence of oxygen metabolism. These ROSes have definite cellular functions. They are involved in cell signaling. However, when they are produced in excess, they can cause severe cell damage and may even lead to cancer. The body is equally equipped to handle these reactive oxygen species by neutralizing them. Examples of ROS include superoxide anions, hydrogen peroxide, hydroxyl radicals, and so on. However, vitamin E acts as a scavenger for peroxyl radicals. Peroxyl radicals are produced when carbon-centered radicals are reacted with hydroxyl radicals. And these peroxyl radicals are the main responsible species for lipid peroxidation. Vitamin E act as a scavenger for peroxyl radicals. They bind to these peroxyl radicals to form tocopherol free radicals and prevent the propagation of these free radicals to damage the tissues. Tocopherol free radical is then neutralized by vitamin C and we get back tocopherol once again. Apart from its role as an antioxidant, presently lots of research is going on about many different functions of vitamin E. To make a long story short, these notable functions of vitamin E include its ability to inhibit and activate different enzymatic activities, its role in signal transduction through modulating phosphokinase C, its ability to control gene expression through NF-kappa beta, and finally its role in apoptosis or programmed cell death. Vitamin K1 was identified in 1929 by Danish scientist Henrik Dam when he was investigating the role of cholesterol in the diet of chickens. He published his work in a German journal and he found that vitamin K is responsible in coagulation, and in German language coagulation starts with K, and that's why the name of this vitamin became vitamin K. Henrik Dam received Nobel Prize in Physiology or Medicine in 1943. It is important to note that vitamin K is synthesized by the normal microbial flora that is present in our intestine. However, the understanding about vitamin K has been changing for last couple of decades. It was previously believed that the vitamin K that is synthesized in the colon are the major contributor of vitamin K for our body. However, recent research suggests that although vitamin K is produced in colon in significant amount, but still the dietary vitamin Ks are the major contributors of vitamin K pool for our body. The vitamin K that is produced in our colon are mainly the menaquinone or vitamin K2 type. The two basic forms of vitamin K that are used in human body are vitamin K1 or the phylloquinone, which comes from the food of plant origin, and vitamin K2 or menaquinone are the type of vitamin K that is produced by the bacteria in our intestine. Both of them have a naphthoquinone ring structure and an aliphatic chain. In the case of phylloquinone, the chain is called a phytyl side chain, and in the case of menaquinone, it's an isoprenoid type of a side chain with varying length. The menaquinones are called MK, and depending on the length of the side chain, they come with a number. For example, MK4 is menaquinone with 4 isoprenoid side chains attached to it. This is the most common vitamin K2 that is found in animal product, whereas MK7 or menaquinone 7 is the type of K2 that is mostly produced by the intestinal bacteria. Food rich in vitamin K include cauliflower, cabbage, broccoli, Brussels sprout, and cereals. The adequate intake level for adult male is 120 microgram per day. For adult female, it is 90 microgram per day. For infant, it is between 10 to 20 microgram per day, and for children and adolescent, it's from 15 to 100 microgram per day depending on the age. Risk of vitamin K deficiency. Average diet does not normally lack vitamin K because vitamin K is present in green leafy vegetables and almost in all part of the globe. Even poor people have leafy vegetables in their diet. And on top of that, vitamin K is also produced by intestinal bacteria in our body. Thus, vitamin K deficiency is rare. However, newborn infants are at an increased risk of vitamin K deficiency because they neither consume green leafy vegetables nor do they have the normal microbial flora in their gut. And also, increased prevalence of vitamin K deficiency is observed among people who have a liver damage through any disease like alcoholism, cystic fibrosis, or inflammatory bowel disease, or those who have recently undergone abdominal surgery. And the logic is same: impaired fat absorption leads to impaired absorption of fat soluble vitamins. The disease symptoms associated with vitamin K is coagulopathy, which is a bleeding disorder. Other vitamin K deficiencies are osteoporosis or coronary heart disease, which was observed to be associated with vitamin K2 deficiency, and vitamin K2 MK7 deficiency is also related to severe aortic calcification. The function of vitamin K is interesting. Vitamin K triggers carboxylation of glutamate to produce gamma carboxyglutamate or GLA. This gamma carboxylation at an important glutamate residue may lead to activation of a protein from its inactivated form. and it has been observed that at least 16 proteins are present in human beings that have a GLA residue for its biological function, and the function of these 16 proteins has been classified into 4 distinct types. Most of the blood coagulation proteins are activated through their carboxylation. That's why vitamin K is important for blood coagulation. It also activates osteocalcin, and thus it contributes to bone health. The matrix GLA proteins are also activated by vitamin K, thus they inhibit calcification in artery, and the Gas6 protein is also controlled by vitamin K, thus vitamin K can modulate cellular growth. We finally come to the concluding part. Vitamin A, E and K are fat-soluble vitamins. Food rich in vitamin A includes fish, egg, milk, dairy products, and green leafy vegetables and fruits and vegetables that are brightly colored. Xerophthalmia is the deficiency syndrome associated with vitamin A. Retinal is the major component of the photosensitive pigment of the rod cells, and retinoic acid acts as a generalized morphogen. Food rich in vitamin E include dried apricot, almond, green olive, vegetable oils, avocado, papaya, cooked spinach. Peripheral neuropathy is the major deficiency disease associated with vitamin E. Vitamin E functions as an antioxidant. It can scavenge peroxyl radical and prevent lipid peroxidation. Other notable functions of vitamin E include its role in signal transduction, gene expression, and apoptosis. Vitamin K is rich in cabbage, cauliflower, spinach, and broccoli. Coagulopathy or bleeding disorder is the disease symptoms associated with vitamin K. Function of vitamin K includes carboxylation at the gamma carbon of glutamic acid, and 16 such proteins where GLA domain is present has been identified in humans, which are involved in various biological functions.